think I did it this week without the having myself echoing in the background. I figured it out finally. So we're going to get started here, and I'm going to start by have a couple of announcements. It's regarding the FINE. First of all, it's nice to see everyone here again for FINE, and it's our final seminar for the Northern Hemisphere Spring FINE seminar series. And I'd like to start off just by, first off, again, introducing myself. I'm Lauren Hayes. I'm one of the three coordinators with Carson Schraden and Eduardo Fernandez Duque. And today I'm hosting the seminar series, the seminar, the last one of the spring. And just a couple of announcements. First, I always like to thank the last speaker. Last week we had Nancy Solomon to give a really wonderful talk on prairie vole social systems. Thank her for her presentation and discussion. And regarding the fall seminar series, we've just about finalized it. I showed this last week, but just to kind of remind you of what we're going to be doing going into the fall. You can see that we have a real nice diversity of talks. We have talks on beetles, paper wasps, carnivores, birds, etc. And there are a couple of additions to the version that I showed everyone last week. Allison Bell from the University of Illinois in the United States will be giving a talk. And Eduardo, do you have a sense of just the basic theme of that talk?
Yes, I mean, Allison has been doing fantastic research covering all kinds of levels of analysis for questions on parental care, focusing her research primarily on frogs.
Oh, very cool. Looking forward.
I'm sorry, fish. I get confused between her and Eva, one of her colleagues. Yeah.
Very cool. That sounds exciting. I'm kind of a different topic. Whoops. My screen just went black. So I don't know what happened there. I will try it again. Can everyone see this okay?
Yes.
Right, thank you. Yeah, so, just a couple other things to point out. One of the things that we're hoping to have in early November is a mini symposium on long-term studies. So we may be reaching out to some of the people that have engaged in this. I think this is a really good opportunity for, researchers to younger researchers. People are starting out to see the value of long-term studies and what kinds of questions we can answer with them. And then there are a number of proponents of taking long-term approaches in this community. So I think it'd be a really fascinating discussion. But this series will be, the schedule will be posted on our website and updated so that you can access it. So when you're away and you're trying to plan your fall Tuesdays, hopefully you can join us for many of these talks. It's gonna be exciting, I think, series. We did want to just kind of reflect a little bit on this semester. We had, it's been, I think, another great season, and we wanted to thank everyone who participates. It's not just about, the organizers and the presenters, it's about the people in the audience participating, and the discussions have been phenomenal. We hope that we can keep that up. And we're particularly encouraged by the emergence of a number of students or early career folks that are joining in the discussion. So we're glad to see that and hope that you can continue this. We feel like the FINE community is well established and connected, and we want to maintain that. So please keep an eye out for any announcements that we have. We probably won't bug you for a few weeks, but we're going to start gathering information from our speakers and making them available to everyone. And we'll start sending reminders about the upcoming FINEs later in the summer. And we hope that we can keep this going for some time. This is going to be our third FINE. We've already started thinking about the spring 2022. And I think we're hopeful we can continue to pursue this further into the future. And, I think the three of us just wanted to acknowledge that, we know that this pandemic isn't over, and that we wanted to sort of acknowledge our friends and colleagues in the Southern Hemisphere, that in particular in the Southern Hemisphere, there's lots of places being affected, but like in Latin America, the rates are pretty high, in South Africa. And we hope that this has been sort of a refuge a little bit from that. And we hope that people in those countries can continue to join us. And we just wanted to know that, we're thinking of everyone there, and we're hopeful that the situation can improve rapidly in the coming year. And hopefully this fine community can help us, stick together a little bit through this really challenging time in our lives. So again, thank you all for being a part of that and making it possible. So with that, I want to, transition into, today's talk. Today I'm hosting Rebeca Rosengaus from Northeastern University in the United States. Becky is an associate professor there, and she has it was really fun to get to know her today. I chatted with her a little bit beforehand, learned how dynamic she is in her career and interests. She received her BS, her master's, her PhD, and postdoc from Boston University, working with, I think it's Mike Trinello. And intriguingly, due to circumstances at the time, she had to finish her PhD writing it from Mexico before she came back to the United States. And that story in itself is really quite fascinating. Becky's research focuses on, termite, particularly termite sociality, the evolution of termite sociality. And in particular, she's interested in how pathogens and parasites play a role as selective forces in complex insect societies. Her work, it takes an integrative approach, which is always interesting to me, and is involved, from her website, it involves sampling in different countries and also in different parts of the United States. And really, her work has an interface between, as I said, it's integrative, so it has an interface between evolutionary biology, behavioral and chemical ecology, and immunology and genetics. And I think that's when I was looking for, to diversify the talk profile that I try and bring in. That's what drew me to Becky's work, was the immunological side of things. I don't think we had anything at the time in this series, so I'm looking forward to hearing about that. She's gotten some funding from the National Science Foundation. She's been involved in REU programs, and impressively, she received a career grant and also a PCASE, which is the top of the funding excellency in the United States. So it's a big accomplishment. My eyes went big when she told me that. That's really impressive. She publishes quite prolifically in good journals, around 50 publications in journals like PNAS, ProcB, the behavioral journals. I think there was Biology Letters in there. And she does all this while having a pretty impressive teaching and outreach profile. She teaches behavior, ecology, and evolution at Northeastern. And I learned today, really fascinating, she's developing a summer course, she's developing a summer course for Northeastern students to go to Israel. But she's also hosted I think it was a remote seminar series on ecology and sustainability and peace building collaboration in Jordan, Israel, and Palestine. And it was entitled Re-envisioning Middle East Ecosystems, Desert Ecology and Environmental Diplomacy, which is really quite fascinating to me. This drew an international audience and really reveals sort of the dynamic nature of her thinking. She's been an associate editor and editor-in-chief at Ecological Entomology, a big journal in that discipline. And as part of that process, she helped develop an editorial apprenticeship program, which the journal apparently allows for PhD students to interact with their advisors. And it turns out their reviews that they submit become a third review. So it trains PhD students in the process of reviewing. So it's really excellent. And so with that, the title of her talk is The Whole is Greater Than the Sum of Its Parts, Emergent Properties of Social Insect Immunity. With that, I'll turn it over to Becky.
Thank you, Lauren. Thank you so much for the nice introduction. Let me see if I can share. Bear with me. You would think I have my act together after all this teaching via Zoom. Anyway, so I wanted to thank all of you, the organizers, for having me here and giving me the opportunity to talk to you about my research. This has been a phenomenal seminar series, and I'm really, truly honored to have been asked to present our research. So what I thought I would do today is, before I get involved in talking about my research in earnest, I thought that it might be a good idea to give you a little bit of an overview of the type of questions we ask and the rationale behind the questions. And then I'll jump actually into the seminar itself. And so I'm trying here to do this. Let's see if it works. So as Lauren told you, I'm actually interested in understanding the different adaptations that social insects have evolved in order to resist disease. And I particularly work with termites. And we know that eusocial insects live under important pathogenic constraints. After all, these organisms are living in colonies that are composed of thousands, if not millions, of individuals in the colony. And these individuals are frequently interacting socially with one another. So the probability of them infecting one another is quite high. In addition, it depends on the eusocial insect, but some of these eusocial insects have nestmates that are sharing a high genetic relatedness with one another. And these insects exploit microbially rich environments. Many of them are foraging in the soil or under the leaf litter. Many of them live inside of wood, and they're even feeding on decayed wood. And so we know that all of these are environments that are laden with potential pathogens. And so in spite of the fact that they live in these microbially rich environments, these guys are thriving, okay? Let me just get the floating thing here off. They're thriving in these microbial communities. And so my feeling is that these guys are doing something right with respect to the adaptations that they have evolved in order to resist disease. And so in my lab, we have looked at parasites and pathogens as posing important selection pressures on anything, but in particularly on the sociality of these insects. And if you think about this arrow here in the middle, as being a continuum in social behavior, going all the way from a solitary or a pre-social ancestor and all the way across, all the way to a eusocial lifestyle, it is easy to envision that prior to the inception of eusociality, parasites and pathogens were more likely already present there at the solitary or pre-social ancestor stage. And these pathogens were probably already exerting important selection pressures on these individuals here. But as individuals and as these pre-social ancestors move through this continuum and cross over what I call this eusociality threshold, I believe that perhaps the adaptations that these individuals here develop or evolve prior to the inception of eusociality then were carried over this eusociality threshold, and the adaptations then were finely tuned and tightly coupled with sociality. And so what this suggests to me is that this is the reason why eusocial insects have very effective adaptations to resist disease within the social context. Let me just go here and put my little laser so I can actually see it. So having said that, in my lab, we've tried to use kind of an interdisciplinary approach. We call this a socio-eco-immunology approach, where we are looking at three pillars: ecology, immunology, and sociality, in order to try to understand more of a holistic tackling a more of a holistic approach to this business of disease and sociality. And so we looked at ecology and we look at how nesting, feeding, and foraging habits predispose these animals to encounter high pathogen loads. We also look at immunology and we look at the mechanisms of physiological mechanisms of disease resistance. So we look at the cellular immunity as well as the humoral immunity that insects have. And then in addition, we're looking at here at sociality. And basically we're asking whether these mechanisms of disease resistance are socially mediated. And so what I'm going to be doing today is actually focusing on these dynamics that we have here between sociality and immunology. And I'm going to be talking today about the gut microbiome of termites and how this gut microbiota seems to be influencing the immunocompetence of not only the individuals in a colony, but actually the entire colony as well. And this is this idea of emergent properties that we will be discussing later on. So the questions that I am going to be trying to show you evidence for that we tried to answer was, can termite gut microbiomes modulate the host's immune response? And so the talk outline that I'm going to give you today is first, the first part is going to just talking about the experiments we ran to demonstrate whether there was a symbiont-mediated immunocompetence at the individual level, whether the microbes in the gut of the termites are actually helping the individual termites to become less susceptible to disease. But then the second part of the talk, I would like to include the termite social interactions, because if you add these two together, what results is a transfer of immune competence, this idea of social immunity across all members of the colony. Again, this idea of emergent properties due to sociality. So in a sense, I'm going to be talking today about symbiont-mediated social immunity, which is kind of a long way of saying we need to talk about the gut microbiome as well as interactions of the hosts in order to understand immunity at the colony level. And so let me first introduce you to what a termite gut looks like. These are the termites that I work with, dampwood termites from California. And it's very easy to pull their gut because you basically hold them here with a forceps on the neck, thorax, and then you get some forceps and you pull very gently and slowly, and you end up having the entire gut coming out. So here you have the crop or the foregut, which is lined with cuticle. Then you have the midgut, which is not lined with cuticle. And finally, here you have what's called the hindgut. It's also called the paunch. And this is very important, and we're going to be talking about the hindgut in a little while. It's also lined with cuticle, and then, of course, you go further, and you have the rectum and the anus, and they poop. And what we see here is that when termites are feeding on wood, they must be also feeding on fungal species, because this decayed wood is loaded with fungi. And so these little oval-shaped things are representing the conidia, the fungal infective spore. They're called conidia. And if a termite feeds on this conidia and it's passing through the gut, when it comes out the other end, in the anus, this conidia has been deactivated. This conidia is not viable any longer. So the question is, what's happening through the trajectory of this conidia through the gut that makes these and renders these fungal conidia inactive? So what's happening there? This is some histological cuts that were done from the gut of a different termite species. But what you can see here in purple is this conidia, this fungal spore that is the infective agent. And what you see here in the little asterisk is the lumen of the gut of the termite. And so this conidium is moving through the gut, and it's not germinating, it becomes inactivated. This is in sharp contrast to what you usually see, which is this here at the bottom. So again, at the top, you have the ungerminated conidia, and I'm going to talk about a little bit later on that. And this here is what you have, these same conidia, when you put them on potato dextrose agar, a nutritive agar plate. However, these conidia love cuticle. Okay, and when they perceive the cuticle and the exoskeleton of a termite or any insect, they start germinating. And so they produce these germ tubes that then invade the host and basically produce compounds and neurotoxins that make the termites become a little bit of a paralyzed syndrome, and then ultimately they end up dying. So again, the question is, what's happened in the trajectory of the gut that makes these conidia not to germinate? And so lower termites are those termites, it's a grouping within the termite phylogeny. Lower termites are considered lower termites if they have both prokaryotic symbiotic associations with bacteria, as well as protozoa, eukaryotic unicellular organisms in their gut. That's what make lower termites lower termites. We also have higher termites, and we'll talk about those later. Higher termites lack the eukaryotic symbionts. They still have prokaryotic bacteria, but they do not have symbionts. So keep that in mind because we'll come back to that later. But the point is, I'm going to be talking today about my lower termites, which are termites that are collected in Western United States in the redwoods. And again, if you take the gut, this is the crop, the midgut, this is again the hindgut or the pouch. If you actually prick that little pouch, what you see here is spilling over of all these protozoa coming out of the gut. And these protozoa are so abundant that they account for 1/3 of the body mass of my individual termites. And historically, these guys, these little microbes that you have in the gut, have been associated with production of cellulases for the termites. So they produce cellulases, and therefore the termite can then digest the cellulose and then absorb all the sugars that are broken down from the cellulose. They're also important in nitrogen fixation and also in the production of some amino acids. And what I'm arguing today and showing hopefully some convincing data is that this prokaryotic and eukaryotic microbiome may have roles that extend beyond the boundaries of the digestive system, that these prokaryotic and eukaryotic organisms are doing something more than just having the termite be well nourished, okay? And perhaps this microbiota is producing antimicrobial compounds that then render the host, the termite, less susceptible to disease. So that's what I'm going to be talking about today. And so we ran a series of experiments, and I'm going to take you by the hand through all of these different experiments to show you that indeed this microbiota is helping the termites be healthy. And so we're gonna, we did some chromogenic gels, and I'll explain what that is, some centrifugal fractionation, in vitro, ex vivo, in vivo, and loss of function experiments in order to test whether the gut microbiota helps control fungal infection, again, at both the individual, but also at the colony level, more very important there. So these are some examples of the protozoa that my termites have in their gut. And you can see that these are the unicellular organisms. They're very, very pretty, and they're actually hypnotizing when you look at them under the microscope. Some of them have long filamentous projections. Those are actually bacteria. So these termite protozoa actually have their own endosymbiont and ectosymbiont bacteria associations. So here you have bacteria that's living inside of the or on the protozoa, and then the protozoa is living inside of the termite, and the termite is living inside of a colony. So you can start seeing the complexity that can be built up, and each of these then have emergent properties as you are talking about the complexity of biological organization. These are other examples. And again, as I say, they're really, really pretty. These are very common in my termite species. And these guys are just fun to look at under the microscope. The termite gut microbiota is not only abundant, it's very diverse, and it's ancient. So there's some very nice studies on amber that have shown that the termites already had this association with the protozoa. This is the amber piece here, and they went in and zoomed in, and they found all of these protozoa that were dated for around 20 million years old. Okay. So at least we can say that termites 20 million years ago already had these protozoa, and these are very similar to what the termites today are carrying in their gut. Even more striking is this find, the amber find, which is showing that the termites already had these symbionts around 100 million years ago. So this is very, very ancient associations and symbiotic interactions. And so the long coevolutionary history that termites are having with their associated microbiota really lends itself to study the nature and the dynamics of these interactions. We know that they're important for nutrition. We have some work that we've done in which kings and queens that do not have their full complement of symbiotic associations in their gut actually cannot reproduce very well. But today I'm only going to focus on the immunity part. And so before I continue, I want to sidetrack for a moment and tell you a little bit about these chromogenic gels, this technique that we used, and you're going to see several gels throughout this talk, and I want to make sure that you know what I'm talking about. So bear with me. This is work that was done by Dr. Mark Bulmer. He's now at Towson University, but he was a postdoc in my lab. And what we did is we worked on creating this technique, and this is, again, the chromogenic gel. Now, these gels are different from electrophoresis, protein electrophoresis, in that when you're cooking the gel, what you're basically using here, in case you're interested in the recipe, you use bis-acrylamide and you add onto that this compound that's called Kirtland. And Kirtland is really a beta-1,3 glucan matrix. Beta-1,3 glucans are the main component of any fungal cell wall. So the cell walls in fungi are made out of beta-1,3 glucans, and this is what Kirtland is all about. And then in addition, when you're cooking this, you're also adding this dye that's called Ramazole Brilliant Blue. And Ramazole Blue covalently binds to the Kirtland. And so you end up having this very nice blue gel made out of this matrix. And then you get your termites and you make extracts of the termites. And for the purposes of this discussion here, I'm showing you all these different casts, but we use these guys here with the little wing buds, and those are the nymphs. And so I'm going to present you basically all the nymph stuff. And if you crush these animals and you make an extract and then you run it in the gel and you allow the gel to run, if the sample has glucanases, then the glucanases will migrate, will sit down on the gel, will digest the beta-1,3 glucan matrix and release the blue dye. And so what you have is you wash this and you end up having what's called clearing zones. So this is showing you a technique that allows you to look at the presence of functional beta-1,3 glucanases because they're functioning. They're basically digesting the beta-1,3 glucan matrix. So they're doing their job, and they are doing it because here you have in the body of a termite without the gut, just the body itself, you see that the termites have only 2 glucanases. But if you run the gut separately, you have multiple glucanases here. So hopefully this makes sense because you're going to be seeing several of these gels in the next slides. We did actually a survey of who has beta-1,3 glucanases across different termite species. And so what you have here is, again, the termites from California that I am working, and I'm going to be showing you the data today. Again, the body minus the gut only has 2, but then the gut has multiples, just as I showed you before. And again, these are basal termites, and these are what we call lower termites here. These guys are also intermediate in the phylogeny, but they also are lower termites because they do have protozoa in their gut. And again, you see 2 only different from these guys here. The species definitely makes a difference in the glucanases. So these guys only have 2 in the body, but they're different from these, and also have a multitude of glucanases, active glucanases in their gut. Then we went to the higher termites. Remember, those guys are lacking the protozoa. And when you run these guys, you have the gut only having 2 as opposed to the multiples, and the body also has 2. And so that was interesting. We even went and got some termites from Africa. Nobody had ever tested even African termites. We were able to get 1 individual, only 1 termite. So this is what we had. And we ran the gut and the body. Again, these guys are also lower termites. And you see, again, multiple in the gut. And I think there was a little bit of bleeding from this into here, but I think that you can see that there's two glucanases here. And here, the ones you see here at the right are the same species as here. So we just repeated that again. And the point is, again, that termites, at least the representatives that we chose through the phylogeny, seem to have glucanases in their gut and also in their body. So what we use these chromogenic gels to systematically tackle questions having to do with the which, the where, the who, the when, the how, who's producing these glucanases, and trying to understand if there's any evidence for symbiont-mediated protection in termites by using these chromogenic gels. So this is one of my students who work in her master's, and there's a reason why she has that mask, and has nothing to do with COVID. I promise you will see why later on. But I just put this on to remind myself to tell you that these protozoa are for the most part anaerobic. Okay, they do not like oxygen. They explode. They basically die and explode if you put them in oxygen. So the gut of the termite is very anaerobic. So we can actually use this to our benefit and exploit this anaerobic nature of these protozoa. And what we did is we put these little termites in this little tube, cotton on one side, cotton on the other, and then we put the whole contraption into these canisters. On one side, we gave them just pressurized air, and on the other side, we gave them oxygen that we know kills the protozoa. And we kept them there in these contraptions for 24 hours. And after 24 hours, we went in and we look at the gut. And those guys that received oxygen really have they did have bacteria, but they did not have any protozoa that was obvious. Whereas these guys, even though they were controls, they were pressurized, they were under the same conditions, they just did not get the oxygen. And these guys were completely phonated, normally phonated. So that's an important technique to use. And we wanted to check whether the defonated termites had the same glucanase activity than animals that were phonated. So this is the phonated controls. Again, you see the body has two, the gut, even though they went through the pressuring and the canister and all that technique because they were controls, you can see the multiple glucanases. But those individuals that were dephonated through the oxygen lost the multiple glucanases here. And so that suggested to us that it is the protozoa, that we were on the right track, thinking that maybe the protozoa are responsible in the gut producing the multiples, except these ones, which probably are termite derived, but all the ones in the middle are probably coming from the protozoa themselves. So once we saw this, we figured, okay, so where in the gut are these glucanases being produced? And so we took the gut of a termite and we basically cut it and dissected it and separated the foregut from the midgut from the hindgut, and the hindgut and the rectum was all one thing. And reminder that here in the hindgut, that's where you see the protozoa. Most of the protozoa and their associated bacteria happens to be here in the hindgut. And when we ran the gels, we were happy to see that only the hindgut actually has all those multiple glucanases. The midgut and the foregut do not seem to have these glucanases, which suggests to us again that we were on the right track and that the protozoa and their associated bacteria are likely producing these glucanases. So who in the gut is producing these glucanases? Is it the protozoa? Is it the bacteria? So we did a little bit of fractionation. We basically took some guts and we smooshed the whole thing into this solution that's called Traeger solution. Again, we had to be very careful to be working fast because, again, we don't want oxygen to destroy our protozoa. And basically we have here in red the lumen bacteria that is free floating in the lumen of the termite. And then we have in blue here represented the protozoa plus the ecto or endo symbionts of the protozoa that are bacteria in origin. So we had all of these and we did first a slow spin, easy to do. And that brings down the protozoa and the associated bacteria. And in the supernatant, we had all the free living bacteria on their own. And so we took the free-living supernatant bacteria and put it into another tube, and then we really brought back down the pellet. So now you have here at the bottom the protozoa with their bacteria, and here you have the bacteria that is free-living in the gut of the termite. And then here you have what we call the cell-free supernatant. Now there's nothing there, supposedly. And we ran again some of these gels. And what we find is, again, that the protozoa plus bacteria pellets that we run have way stronger signal of beta-1,3 glucanases than the lumen bacteria itself. There's a little bit there, but really not much comparatively speaking. You can see that the pellet here is huge. And these, of course, are the regular controls. We washed the gut, and that just shows to us that we were not very good washers of termite guts. But the pellet really has a lot of this glucanase signal. we then figure out, okay, let's try to grow these guys outside of the termite and see if we can do some ex vivo culturing of these gut microbes. And it's, again, not easy to grow these symbionts because, again, not only do they not like aerobic conditions, but they really need a lot of special conditions to thrive, culturing outside of the insect. And so my student that I showed you before with her mask decided to go and get this fistulated cow close by here to Boston. They have these cows in this agronomy school where they have this hole that goes all the way to its gut. Who would know that these people are doing this? But I did not know that until she came up with this idea. And basically, she shoved her hand into this stomach and took out a lot of these juices and the cellulose that the cow has in her gut, and she brought it back to the lab. And so that's why she's wearing this mask, because this thing stunk awfully. It was terrible smell. We couldn't use the lab for weeks after this. But what she was trying to go for is this green liquid stuff, because apparently, from what we had read, this green liquid stuff is very conducive for the sustenance of this particular protozoa. At the end, I have to be honest, we gave up on this because the smell was just awful. And so instead, she found yet another technique that worked really well for us. The point is that she took this gut, smashed it out into this other solution. It's called Traeger solution, and she left it anaerobically for 12 days. And what you can see is that this solution has, again, these beta-1,3 glucanases, which shows that, again, when you take away and grow the protozoa outside of the termite, you're still getting this signal of these glucanases. And when we looked at what was in there, we basically found this little guy being very abundant. Maybe others are producing these beta-1,3 glucanases, but maybe they just didn't do as well. But at least this guy seems to be associated in a 12-day-old culture, ex vivo culture. This guy was very prevalent. So this is a Trichomatopsis, and it's, again, a symbiont that lives in the termite. So perhaps these are the guys that are producing the glucanases. So how are these glucanases working? I've shown you that the glucanases are there, but we don't know if these glucanases are actually helping the termites or not, or whether they have any antifungal activity. So what we did is we used, again, Metarhizium, which is the conidia that I showed you before that was germinating. This is a photo of the conidia on the cuticle of a termite. So this is a little spine, a little hair on the termite. This is the cuticle here, all this pitted area. And this is the conidia that stick to the cuticle. Remember, these conidia love cuticle. And so what we did is we did two experiments. One of them, we took the guts of the termites, we made an extract, and then we combined this with conidia by itself. Or we took the body of the termite without the gut, we also smooshed it, did an extract, and also incubated that with conidia itself. And then we plated this conidia and we counted colony forming units. We counted how viable this conidia happened to be. And so this is the methodology we use. This is the conidia by itself. And after you plate the conidia, you can actually get these colony forming units. And so at the top, I'm showing you the control. So basically this is conidia with acetate, but no gut, anything, just the conidia and the acetate. And you see that the conidia is growing nicely. We also incubated this same conidia with the extract of a single gut or a single termite, and look at the difference. This is the control again, and this is the conidia that was incubated with one single gut. And what you can see here is that only 2 colony forming units grew, whether this is a control. So clearly these glucanases, or this gut stuff that's happening in the gut, the extracts themselves have potent antimicrobial properties. We also wanted to quantify this a little bit further and actually check for differences between different colonies. So this is the same research, but now I'm showing you the quantitative data. On the y-axis here, you have the median number of CFUs, the colony forming units, as a function of 6 different colonies that we tested. And in the different bars, the blue is the gut extract. Again, we dissected the gut, we ran the gut, with conidia, and we looked at the germination rate. This is the body minus the gut in green, and this is just the control, the conidia by itself with the acetate, but no gut or no termite. And what you can see is that consistently, every time that we run the gut across all the different colonies, this gut incubation with conidia really reduced conidia viability quite dramatically, significantly so. The body was a little bit of a different story. It was not always consistent. So here you see that the body, the green here, is not significantly different from the control. Here, on the other hand, you see that the body is reducing conidia viability relative to the control. And in this weird situation, I don't know what happened in colony D, but you actually had better germination when the body was there. So it's not so sure what's going on with the two glucanases of the body, but clearly with the multiple glucanases in the gut, you definitely get antifungal activity. Now, the question is, what if you block this enzymatic activity? What if you actually block the beta-1,3 glucanase activity? And luckily for us, we were able to find a compound that basically binds to the active site of the glucanases. And this is called gluconolactone. I'm going to call it GDL for short. And GDL, we know, blocks effectively the beta-1,3 glucanase activity. And the way it does, and with the way we demonstrated this, is by basically using the same sample and loading it on these two gels. The only difference between the top gel and the bottom gel is that the top gel was incubated after running it, it was incubated in also gluconolactone. So it was being washed and the enzyme activity was being blocked at the top, whereas the same samples run in without the gluconolactone were present here. So again, you can see here the gut of the defonated termite with all the different types of glucanases, whereas here we don't see it. So we know we can deactivate and render with GDL, we can render the glucanases inactive. So if you block the enzymatic activity, what would you expect of conidia viability to happen? Well, you would expect that conidia would be rescued. And so this is the research that we were trying to see if in fact the beta-1,3 glucanases in the gut produced by the protozoa are actually responsible for the antifungal activity that we saw ourselves. And so what we run is, again, these are the results of that experiment. On the y-axis, again, you have median percent germination, and we had different treatments here. The stock here is just the stock's conidia by itself, and you see that it has very nice germination rates. The control here is the conidia with the acetate, and we know that the acetate also is not really reducing significantly germination rates. We ran a different control where we added just the conidia with GDL, and you see that GDL also is not itself hampering or reducing germination. Then we run the gut extract normal or the body extract normal, and we see that these guys significantly lower germinations than your controls. So again, this is very similar to what I showed you before. And in the next one here, you in green, you have the gut extract incubated with GDL. So the enzymes are really inactive, and this is inactive here in the body. And you can see that now you have rescued conidia viability again. So clearly the fungal activity that we saw in our plates may be explained by the beta-1,3 glucanase activity that these guys are having in their gut. So just to recap, where are these beta-1,3 glucanases? Well, it depends on the tissue that you're running. If you're running just the body itself, you only have 2 glucanases. But if you're running it in the hindgut, then you have multiple glucanases. Who is producing these? Based on our fractionation experiments, it seems that it's more the protozoa and its associated bacteria. It was very hard to run a project where we could divorce or separate the protozoa from its own bacteria. We kept on killing everybody, and so that was impossible to run. So we decided to just keep it as, at least we know that it's protozoa and its associated bacteria responsible for the production of these glucanases. How? Well, it seems that the glucanases have important antifungal activity. And the next question is, are these termites benefiting from these beta-1,3 glucanases? We've demonstrated a lot of good stuff, but we don't know whether this translates into the termites actually doing better when they are coping with mycosis, with fungal infection. And so for that, we asked Ala, an undergraduate, to run these in vivo experiments. She used, again, the same technique of pressurizing the termites and oxygenating the termites to create defonated termites on the right and phonated termites on the left. And so we had around 180, 190 individual termites per treatment. Some were controlled, some were exposed to these 10 to the 5th conidia, so this kills the animals. And then we ran their survival curves. We follow survivorship for 21 days just to see who is dying and when they're dying. And so these are the results of those experiments. On the y-axis, you have percent survival of the termites as a function of their different treatments here in the symbols. And here you have days post-exposure. So again, we ran this until day 22, 21, 22 days. And so what you have here in blue at the top is those animals that were the controls. So these guys were phonated. That means they were exposed to the pressurized air, but they did not have oxygen added to it. And they were exposed to suspension that was lacking fungal conidia. And so there's no reason why they should have died. And they really didn't die much at all. Then we have the ones in red, which are those termites that were dephonated, but were also exposed to the suspension lacking fungal conidia. And what you do see here is a steady decline in their survivorship. These guys should not have died, but I think that we are assuming they should not have died of mycosis because we were not exposing them to the fungus. But we think that they're dying because of malnutrition. Remember that we got rid of their gut microbiome, and so now suddenly they don't have their gut microbiome that helps them digest that cellulose. And so perhaps at the beginning there's really no mortality, but then as time progresses, I think that these differences here get bigger and bigger because of malnutrition. The interesting comparison that I want to have you focus on is this one here. So in the open symbols, you have the phonated termites that were exposed to a pathogen, the fungal pathogen, and they are dying relatively faster than any of the two here. Here you have a big drop and then it keeps on going. And it's interesting to see that these guys here have better survival than their counterparts, which were dephonated and also exposed. And so clearly these guys are dying very, very fast. And so there seems to be a benefit of having an intact microbial community, because when you have this full complement of your gut symbionts, you don't only do better food-wise and nutrition-wise, but you do better at coping with fungal infection as well. So we also wanted to look at when during development are these glucanases showing up. And so we took some first instar larvae, the baby, tiny babies that just hatched from the egg. And you can see already that there's a little bit of signal there in these glucanase gels. When you go to the second instar, you see a lot of more signal. And so what's happening here? And we think that what's happening is that the parents or the nestmates are basically defecating. They're producing this droplet that's called Proctoredial Feedings. This is a very common behavior that termites do, where they are sharing their poop with one another. And this droplet here is loaded with symbionts and is loaded with beta-1,3 glucanases. And what we think is happening is that the parents or the older nestmates are vertically transphonating their young siblings or their babies, and they're passing these beta-1,3 glucanases along with the proctoredial feeding. And so these individuals here who are born without protozoa are getting the protozoa from the parents or from their older nestmates, and then also getting the full complement, not only of the protozoa for nutrition, but the protozoa for dealing with fungal infection. So this is where the social interactions between parents and offspring, or nestmates with younger nestmates, are coming together to allow for the spread of this kind of protection from fungal infection to pass to other individuals in the colony. So this is probably something similar to social transfer of immune capabilities, which is pretty cool to think about social insects engaging in the sharing of immunocompetence. Just so that you believe me, I hope that you can see this. Let me just escape for a moment here with this thing. And hopefully you can see, this is a droplet of that poop that I just showed you in red. So this is true poop under the microscope. And just to show you that this poop is not only excrement, it's not only poop. And hopefully you can see it moving. I don't know if you can see the droplet moving. Someone can tell me yes?
Yes.
Okay, great. So it's moving because it has this protozoa. Okay, the protozoa are swimming around, twirling around, and as they're swimming, then they're being passed onto the gut of the recipient. And so what you have here is basically a technique by which these termites, through their social interactions, now it got stuck. Through their social interactions, are passing immune function from one individual to another, a passive transfer of antimicrobial compounds that then allows these recipients to be less susceptible to disease. These glucanases can be passed probably both from the termite itself or the termite and the microbial community that they happen to have. We're not really sure, but it seems that the liquid feces that I just showed you, we ran just the liquid itself, and it's loaded with glucanases as well. Okay, so these are the glucanases here of the droplet that they're feeding. So termite poop is very, very important. I know that I'm getting close to be done. So termite poop, again, is important for termites in that you can see two different types of poop. You have the droplet here that they engage in proctoredial feeding, and they also defecate these solid pellets. And they're constructing their nests and they're paving their galleries by adding and using these liquid feces to cement the fecal pellets. And between the fecal pellets and the cement here, basically they are lining their galleries, and we shouldn't be wondering why fungus doesn't really grow in these colonies. This is loaded with fungus. It's just it doesn't grow because the termites are using their poop in order to pave their inside of their nests. And so just to go back and just say that we think that, I'm going to go very quickly here, but this here may be actually passing on immune competency to the offspring, and these guys are then receiving proctoredial feedings that probably are boosting immunocompetence at the colony level. Okay, so what's important here for me is just to finish saying that I think we need to keep track of the holobiont, meaning not only the individual termite, but the individual termite with all its microbiome together, and of course, all its hologenome, right? Each of these organisms, microorganisms, have their own genetic makeup as well. All of it, the whole termite with everything inside of it, should be considered the unit of selection in evolution. And this is my final graphs, basically showing you that the system can be very complex. You can have viruses that live inside bacteria, bacteria that live inside of protozoa, protozoa that live inside of a host, and that each partner in this particular setup has its own interests, and that the interests here may not necessarily be aligned with the partners, right? Each individual here, the virus or the bacteria or the protozoa, have their own interests. But the point is that when we look at the end product of natural selection, we have to take all of these guys into account when we look at the benefits or the fitness related benefits. So in termites, not only do we have the termite host here, but now we have the termites living inside of a social insect colony. And so this is where we can see these emergent properties going up the complexity in social organization and biological organization that allows us to understand that insect immunity can be the result of amalgamation of many units and many different species all living inside of one another. And so the take-home message again is that termites have a diverse gut microbiome. These microbiomes are known to, we show, hopefully I convince you that they're important in reducing disease susceptibility at the individual level, but because they are engaging in proctoredial feeding and social interactions with one another, they're passing immune competency from the individual to another individual. And so you end up having colonies that are all really happy. They're putting all those glucanases also in their nest. And so the entire colony then is benefiting from this individual immune competency that is actually coming from the gut microbiome that they have inside of them. So it's a complex system, but I think it was a very fun project to work on. And so with that, I just want to thank everyone who was involved. The students were amazing, and as well as the National Science Foundation for funding this research. And thank all of you for coming over to the last seminar. Again, guys, you did an awesome job in this seminar series. I really appreciate being here.
Thank you, Becky. That was really fascinating. Really cool to hear about something totally, I have to admit, totally outside my realm. So that's really cool. See how it associates with sociality. So this is a time when we have an opportunity to engage in discussion with Becky about her work. First, I'm going to tell people over YouTube, if you have questions, please type them in the chat. I'll be monitoring that and transferring them over to the folks here. For the folks on Zoom, please type a question mark in the chat, and I will call on you as they come in. And please, when you ask a question, remember, please say your name and where you're from so we can all get to, continue to get to know each other. Okay, so so the first question will come from Miles.
Hello, just giving me
Hello.
a second. I'll make sure my screen here isn't too foggy so you guys can see me a little bit clearer. My name is Miles Machinsky. I'm a second-year master's student in Lauren's lab at University of Tennessee in Chattanooga. And fascinating talk, really exciting to hear about this whole study system and the different symbiotic associations that you guys are aware of or have gotten to study in the termites. I guess my questions are centered around maybe what you think your next steps are. So while listening to your talk, I couldn't help but think of some ideas for like maybe questions that you could ask next or different studies you could do next. The first one would be, since there are so many, there's a high abundance of different types of protozoa and bacteria within the whole termite ecosystem, is there any plans to, or let's back up, do you know of any characterized protozoan species that may be present in the gut? And then are there any plans to like run like a qualitative PCR and see how abundant certain species are and kind of like who's there? Yeah,
so there's been some studies, particularly I suggest if you're interested in this, European colleague Andreas Brun. He is very well known for all the research that he's done on the protozoa of the termites. So there's a whole list of a lot of identification of these little guys. And again, these are obligate at this point in evolutionary time. These guys only can live inside of the termites, and the termites cannot really live without them. And so it's become a symbiotic association that is an obligate association. And I think that that he has done a lot of identification. To be honest, we were running other projects as well, which I can touch and talk about a little bit, that took my attention away from the gut microbiome. And instead, I was more interested in another very interesting research project that my graduate student had, where we were talking about transgenerational immune priming, and whether the queens and the kings can actually be immunized, and whether they can then create an immune response and then pass that immune signal or immune function, perhaps as prefabricated proteins or perhaps as an immune elicitor onto the eggs before the eggs are laid, even before the eggs are fertilized, so that when the eggs are oviposited, the eggs already have some kind of a signal saying, beware, there's pathogens. The parents exposed, the parents already experience some pathogenic challenge. And so the mothers are imbuing their eggs with a signal that tells them, be ready because there's pathogens out there. It's like a little present that the mothers are giving to their progeny. And so we moved a little bit away from the gut microbiome and into this idea of transgenerational immune priming. And it's been a tremendously fun project as well. So
I bet, yeah.
We haven't really looked much into that thing with the microbes, but one of the things that I would love to get involved, but I'm not a molecular biologist, and to be honest, I am kind of a little bit of scared to get into that area, is if you remember the gels, you had two in the body of the termite, you had two glucanases that when you run the gut, they were also in the gut. And my question in my mind is, how did these two glucanases in the tissue of the gut, not the protozoa, but the tissue and the termite itself, got there? And I'm thinking that there might have been in evolutionary time some kind of lateral gene transfer from the microbial community into the genome of the termite, so that the termite body now has these two glucanases. If I could get involved in doing that, if any of you want to run this for me, I would prepare for you all the termites in the world, but it would be such fun to see that there was lateral gene transfer from the protozoa into the tissue of the termite. So that would be my favorite thing to do and test now.
That's really exciting. Yeah, that sounds like a great second route to take as well. And what you were explaining before about looking at the, I guess, transgenerational immunity is really interesting too. That I'm going to wrap up here, but that kind of led to my second idea. And I have a question. Can you separate the baby termites? Like after they've hatched, will they be okay to survive in an enclosure or something on their own?
No, they really need to be transphonated. Otherwise they don't live. So they really need the society to take care of them, to groom them, to lick them, to feed them, to give them proctoredial feeding. The first and second instar, which are tiny, 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 the size of the egg, really. When they hatch, they really need to be taken care of by the nestmates or the parents if it's a colony that is just getting established. But all the research, a lot of the research that we did with transgenerational immune priming required that we would take the king and the queen, pair them. So we played matchmakers and we put a king and a queen and make it really romantic. And they made it inside of this petri dish and then gave us the eggs. And so we could then go to the eggs, and we're delighted to see that the eggs of parents who saw or experienced the pathogen, and in this case we were working with bacteria pathogenic pressures, who saw a bacterial pathogen, the eggs themselves have two genes that seem to be upregulated. Two immune genes are actually upregulated. One of them is called relish, and that was significantly upregulated, more than twice the amount of transcript in the eggs of parents who saw the bacteria relative to the controls. And the other gene is called IMD, which is very important in the immunity cascade of the insects. And that was also upregulated, but not significantly so. But at least we see some changes in the transcriptome of the offspring based on the parents' experience of pathogenic constraints. So it's pretty cool.
That's really exciting. Awesome. Thanks for sharing that.
Thank you for the question.
Okay, our next question comes from Karsten.
Yes, hello, my name is Carsten Schradi, and I'm a researcher at CNRS in Strasbourg, France, studying mammalian social organization. Thank you. That was really a fascinating talk. And
uh-oh. Can you guys hear?
We could. I think Karsten froze there. I'm going to come back to Karsten's question when his situation is resolved. I have a question from YouTube that I can ask.
Sure.
So Max Verhey Says he's a grad student in Wageningen University, and he was wondering how general the process is you describe, especially since you focus on one specific fungus.
That's a great question, and I agree that that's not necessarily what I should be doing. I think that it's important to actually maybe run some of these experiments with a different type of fungus. There's a lot of different entomopathogens, and yeah, I agree with you that I'm not sure. I don't think anyone has actually tested these. We've tested other termite species, and they seem to be, again, including higher termites. So those that are lacking the protozoa, when they do have 2 glucanases, and those 2 glucanases in their body and in their gut seem to be doing a very good job as well. But that's using the same fungus. So I agree with you that maybe we should expand this and actually test with other fungi as well. It's important, yes.
Hey, Max, if you want to have a follow-up question, please just type it in YouTube, and I'll come back to it later. So I think Carson is back. Carson, do you want to ask your question?
Yeah, sorry, I will try. I was kicked out. And maybe you just replied a question while I was gone, and I know you gave the answer twice during your talk, and I was just too stupid to follow up. Can you explain to me what is really I got the difference between the lower and the higher termites with the protozoa. But can you tell me, where does this terminology come from? Why are the ones called lower, higher? It implies something like evolution. When did they diverge in evolutionary history?
I agree with you that this is probably not the best. I don't know who came up with this, coined these terms of lower versus higher termites. I think that the issue is a historical baggage. Most of the basal termites do have protozoa and bacteria. As evolutionary tree starts progressing and moving more towards more derived species, somewhere along the way, the derived species ended up losing the protozoa. And we're not really sure how this happened or when exactly this happened, but the protozoa were lost in the more derived species. The more derived species are the most probably abundant. I mean, termites are very common, but the lower termites have many families in the phylogeny. The higher termites have fewer families, but they're very, very numerous. These colonies are huge. The colonies, as you get more derived, these are the big fungus growing termites, and many of you have worked in the South America, where you see the carton nests on the trees. These are huge carton nests, and those are higher termites. I think that originally people assume that if you were a primitive, basal termite, then by definition you had protozoa, whereas the higher termites that are more derived should not have any protozoa. And so I think this is just it coined historically, but yeah, it definitely gives the impression of an evolutionary pattern that does not necessarily hold. So yeah, maybe we should change termite people, but it stayed there. And how the protozoa got lost is not really well understood, but it came about the same time as when termites became arboreal, for example. So termites, most of the basal termites, those lower termites that have protozoa, are soil dwellers, are eating wood, and they eat decayed wood and live inside of the wood or nest in the soil. But as termites derived more and more and became more phylogenetically advanced, then you have these animals moving, or these colonies moving arboreally, or starting to create these huge mounds. And so that's more of maybe it's an ecological shift that then allowed for them to lose the protozoa, and they did not need the protozoa because now they're still using cellulose, but somehow they can deal without the protozoa themselves. And today we know that the termites can also produce cellulases themselves. So it is not the case that only the protozoa are responsible for digesting the cellulose that the termites are eating. The termites themselves have genes that are cellulases, that produce cellulases for them. So it's probably a combination of both cellulases from the protozoa, but also of the termite itself.
Okay,
I don't know if I answered your question.
Okay, our next question comes from David Valencia.
Yes, hello.
Hi.
My name is David Valencia. I am studying a master's degree in Université Sorbonne Paris Nord in Paris. I wanted to thank you. It's a really good talk. And I wanted to ask you about the defonated termites, the one that you remove the protozoa with the oxygen. If it's possible to restore the fauna by giving them the watery feces that the larvae eat?
Yes, the short answer is yes. You can defonate them and then actually phonate them again by milking the liquid poop from the donor that is healthy and has a full microbiome, and you suck that up with a pipetter, and then you put it on paper, and the termites will eat the paper or drink it. You can actually give it to the termite, and the termite will get some into it, and then you can actually phonate them again. Yes.
Okay, thank you very much.
Okay, our next question comes from Madison Roberts. Madison, are you still out there? There you are.
Okay, I'm Madison Roberts. I am an undergraduate at the University of Tennessee at Chattanooga, and I'm in Lauren's lab. I was wondering, could you elaborate more on termite, I guess, colonies, if you call it, and their interactions? Like, I'm imagining them similar to bees or ants. Like, is there only one queen, or who's all mating, or who's taking care of the offspring?
Yes, great question. They are very similar in organization as, for example, ants, in that they have a queen, but termites differ from ants or bees or wasps in that they actually have the king as well. So you have father and mother coming together in a monogamous mating system, starting a new colony. And the father is a really good father. He really does a lot of the work along with the female. So not only when they come together and find each other, they will, the female, they fly. I don't know if you remember the photo I was showing with the different casts. There were some individuals that had wings. Those individuals are swarming. So the colony produces swarmers, or what we call alates, winged individuals, once a year, and they fly. And the female then lands somewhere and she raises her butt up and she produces a pheromone. And the pheromone then attracts the male. So the male starts, finding the female, he lands next to her. And when the female feels that the male is just behind her, touching her with the antenna, she starts running around. Sometimes they break their wings before, sometimes they break their wings after, but they basically have a little suture in the back of the wings. And when they're ready to start walking, they will press their wings down on the ground and it clips. So now you have a termite that lost the wings, a king and a queen that lost the wings. They're still virgin and they're moving around finding a good place to nest. And that includes some kind of wood that happens to be at the right decayed stage of the wood. They make a little hole in there, they go in, and then they close themselves again with these feces that they're producing. So they basically make what's called a copularium. So they excavate a little hole where the two of them fit in there barely, and then they lock themselves in. They start grooming, being very lovely to one another. They will mate in there, and that's when they start laying. She starts laying the eggs probably around 15 to 20 days after they got together and started the colony. And so, and then the male is as useful as the queen. He grooms the eggs, he licks the eggs, he helps excavate the nest, he enlarges the copularium, he's grooming the female is grooming him. They're very, very useful. And we have some work that shows that a female without a male has no chance. So he is doing a lot of work with her. The female has to do a lot of things alone. She can not only produce the first batch of eggs and do the copularium and do the cleaning, and they're feeding on low nitrogen food. Cellulose has really little nitrogen. So they need to have the help of the male in order to, be successful in establishing a new colony. So the males are very good. Not only that, but the termites are diploid. So you guys probably remember your classes of the social Hymenoptera, the bees, the wasps, and the ants, the eusocial Hymenoptera. Those guys are haplodiploid. So the male is haploid and the female is diploid. Termites are diploid. Both the males and the females are diploid. So that's a huge difference between the genetics of sociality, with respect to how come these diploid individuals evolve sociality, when so much was said about, haploid-diploidy being important in the selection of eusociality and high genetic relatedness. These guys are little families at the beginning, and then they just grow and grow and grow, and everyone stays behind and enlarges the colony. Did I answer your question, Madison?
Yeah, that was really fascinating. Thank you.
Yeah, they're fun to work with.
Great. Our next question comes from Zulema.
Yeah, hi, Zulema Tang Martinez, University of Missouri in St. Louis. I'm retired, and thank you for a fascinating talk. My question actually has to do with one of the papers that you sent for people to read. And I just looked at it very quickly, but as I remember, it's a paper in which infected termites, I assume faunated adult termites, were either kept in isolation or in a social group, and the ones in the social group survived better than the ones in isolation. And I'm wondering if the proctoredial feeding could also have been relevant there, or if you have any, feedback on that.
Yeah, this is interesting because I was actually just asked here by the university, people are talking about COVID, and, we need to be isolated, right? Human societies need to isolate themselves and not interact with one another. And here you have the termites that are actually doing the opposite, and they do better when they're in groups than when they're in isolation. And if you expose a termite to a fungal pathogen, like the one I talked about, not only will the individual that's being exposed respond to the pathogen, but it will elicit the behavior of other nestmates to actually groom it. So allogrooming is very, very important in preventing the establishment of disease. And we have some, maybe that's one of the papers that you read. If you look at some of the graphs there, when you do a grooming behavior, it increases dramatically. So the termites not only perceive that they themselves have been exposed, and if they do, then they start walking funny. You can actually see them being very uncomfortable. This is the only word I have, is the termites look uncomfortable as they're walking over the fungus. They kind of tiptoeing. And they start shaking. If you give them a high dosage of fungal conidia that is lethal, within 5 minutes, and I can show you a video if you guys want me, I can go and try to find it and show you the video. But these guys start shaking. It's almost like they have a seizure. They go forward and backwards, up and down, all at the same time. And they continue shaking until they die of infection. And the reason we have some work that has demonstrated that this is almost a pathogen alarm response, and it's a dosage dependent response. If you give them a lethal dosage, they will shake like crazy, and the vibrations that they are producing on the ground basically tell nestmates far away not to come close. And so the nestmates stay away. Those nestmates that are confined with that individual then change their behavior. They can't go away, but they can start grooming one another quite a bit. And you have a termite that has been exposed, and she sits there and turns herself around so that the other ones are licking it with their mouth parts. And they go from head to toe, you know, all the way to the anus, and she turns her belly around to be cleaned and cleaned and cleaned. And I think that that's part of the reason why animals that are in groups are doing better than those that are isolated, because those that are isolated cannot groom themselves. Ants are very good at grooming themselves. Ants are very flexible. They have a head, a very tiny thorax, and then the abdomen. And so that allows them to bring their abdomen forward and sideways, and they're able to move a lot. Termites have a head, a very thick, wide thorax, and a big, bulky abdomen. And so they really are not so flexible. So they rely on the others to groom their body, because these guys can only basically clean their antenna, maybe the front legs, and that's about it. So the others are coming in and helping in the cleanup. And definitely grooming, I'm sure that proctoredial feeding may have helped there. When the dosages are so high, you kill them anyway, because the fungus is so nasty. But definitely there's social, that's when I'm talking about socially mediated disease resistance. This is one of the things that I'm talking about, is that they changed their behavior, and their behavior was probably tuned, highly tuned, and to living in societies that allow them to have allogrooming, a very important adaptation to resist disease at the group level.
Yeah. So to follow up, unless my dog starts barking, in which case I will turn off the microphone. So when they're infected with a very high level of infection, they start shaking, and other termites don't come close.
Mm-hmm.
So when the licking occurs, is that because they're confined together and so the other termites can't stay away? Or is it because it's a low level of infection that has occurred, and so the licking is able to help?
The licking helps even if it's a small, a low dosage. So you're right,
Okay.
And the setup that we have is we either confine them and then allow the other ones to try to come in. And if you allow them to try to come in and actually do the grooming, they don't come if they're far away. But those that are being exposed at the same time with the individual that's vibrating, they are going to stay and lick their nestmate.
Right.
So it's a dosage dependent kind of a thing. They somehow perceive the difference between a lethal dose versus a non-lethal dose. And a non-lethal dose will elicit grooming from others, but they don't really start shaking, and everyone is grooming one another fine. It's the shaking happens only when you hit a magic number of 10 to the 7 conidia per mil. If you give them a 10 to the 7 conidia per mil, they start shaking like crazy. If you give them a 10 to the 6 or 10 to the 5th, they will not shake, which is weird. I don't know how they perceive this, but they do.
Okay, thank you.
All right, so I'm going to jump over myself and next, Alexa to ask the next question, please.
Hi, my name is Alexa, and I'm a PhD student at Yale, and Eduardo is my advisor. So I'm going to anthropomorphize for a little bit here, but when they're like kind of doing these like fecal transplants, like they're kind of consuming their fecal matter, the ultimate cause seems like they're exchanging this microbiota, and that increases their immune function. But is there kind of a proximate cause for that, or a behavioral reason? Like, are they getting nutrition from the fecal matter, or like, could they be bonding in some way? Like, why are they kind of doing it on a individual basis?
I think that this proctoredial feeding may have originated, and again, I'm talking and here, I'm making this up as I'm going along here, but I think that the termites may have been selected to share with one another. Feces usually has nitrogen, right? That's why we fertilize our grounds with, cow poop and so on. So that feces has a high content of nitrogen that the cellulose they're eating does not. A lot of that nitrogen is coming from the protozoa. A lot of that nitrogen is coming from nitrogen fixing bacteria that live in the gut of the termites. And so I would imagine that originally the reason why termites started engaging in these fecal movement from one individual to another was because nitrogen is such an important compound that you need for almost anything, right? You need to create new protein. If you're just born, you need to grow, you need to mold, you need to create a heavy sclerotized cuticle, et cetera, et cetera. So you need nitrogen for immune system. You need nitrogen for basically any function, create your enzymes and so on. And so by you getting the nitrogen in the poop of your nestmate, you probably were doing better. And you could then grow and develop normally and use that nitrogen for other reasons. So perhaps the original reason for proctoredial feeding was the need for nitrogen. But then, you know, you get added benefits sometimes of and pre-adaptations that allow for this transfer to play more than just one role. So now you're not only passing nitrogen, but now you're passing beta-1,3 glucanases along with, you know, perhaps pre-digested wood that then the little ones that are born can use for growing. It's easier for the recipients that are babies to kind of have pre-digested food through the feces of the nestmates, and therefore they can easily digest that better. So I think that this is, you had an original reason for it, but then serve as a pre-adaptation to get other benefits added to the system. Why are they doing it? I think that this is, you know, I have an evolutionary response, as I just said, an ultimate response. Why do they do it individually? So I think selection actually fostered this as an evolutionary solution to the problem of low nitrogen in their diet.
Yeah, that makes a lot of sense. Thank you. That's interesting.
Yeah. There's a lot of animals that eat poop. I mean, you have horses that eat poop, anything that's using cellulose a lot, and they really don't have these guts where you know, like cows, that you have multiple guts and multiple stomachs and all these protozoa feeding and helping you break the cellulose. Many animals will actually eat poop, including my dog, unfortunately.
Yeah, no, I mean, it can be like a valuable food resource. But I guess if it really is just simply for the nitrogen, then you could just be eating your own fecal matter. And like the exchange is kind of a different, you know, an added element to that.
They do eat their own poop as well. So it's not only that they just poop and not eat their food. Sometimes I've seen them pooping and then turning around, and just the way they just put it on the floor, that's what they're eating. So they will eat their own poop. And they will cannibalize other individuals in the colony. So if nitrogen goes really down, they'll actually engage in cannibalism as well, eating live nestmates that are then probably helping them replenish their own nitrogen, including the moms will do that, or the fathers. But in general, they're really nice parents. I love watching them behave towards their babies. They're really good papis and mami.
They're really nice unless they eat their children.
Well, you know, aren't we all nice until we eat our children? Mamas do that too.
If there's anyone else who has a question, please throw it into the chat. In the meantime, I'll ask a question. And again,
Sure.
I come from ignorance about this system, and I guess my question, it relates, and I'm not even sure I can formulate a great question, but it relates to development. And now these guys have caste systems, right? Is there any relationship between the development and access to this poop? Do we know anything about this?
I think that everyone has access to the poop. The only individuals that I particularly think will have a more difficult way of getting the feeding is the soldiers. So I don't know if you saw the photo again. The soldiers are massive. In a big colony, the soldiers have these big heads, very sclerotized heads with these very long mandibles. And so they have a hard time to actually turn their head in a way that allows them to eat or drink the poop from a donor. So I've seen them munching on fecal pellets, but I've never seen them drinking directly from the poop of the donor. The little guys, the babies that are just hatching, are running to mom and dad and actually getting this from they touch the end point, and they will, mother or father will produce this droplet. Everyone else is as comfortable doing that as well from one another, nestmates to nestmates, younger to older to younger, and vice versa. The soldiers would be the ones that have probably a physical hardship because of their big mandibles.
And does, I guess what I'm wondering too is does, again, I'm not quite sure how to ask it, does the development of the young, is that impacted by sort of the quality of how well they keep the nest, or sort of what's actually being delivered in the poop?
We don't know.
Okay.
We have not really looked at that. I can tell you that the colonies in general, and even if we've set up, I don't know, millions, I want to say, perhaps, I don't know if millions, but many thousands and thousands of baby colonies, we call them. So we get the king and the queen, we pair them, we put them in petri dishes, and even if you give them everything that they need, if they need water and everything's clean and everything's, the wood is just right and everything's fine, we still get 60% of the incipient colonies dying on us due to whatever, fungi, bacteria, stuff that grows there, who knows. So even 60% of the colonies died, even when you do everything right.
Mhm.
So the colony develops very slowly. These guys are very slow growers. Again, they start laying their eggs, but their first egg, or the first few eggs that they lay, take 2 months to hatch. It's not a fast hatching, it's other species, the eggs are a week and then they're out. Not these guys. These guys are really slow growers, slow developing in the embryo. And it takes several months for these embryos to actually hatch. And then you have this tiny little thing that everyone steps on, in the colony. They groom it and they take care of it. Sometimes they eat it, but they don't really care much about the little tiny ones. As they're growing older, then you start seeing more attention and more delicate attention to these young individuals. I don't know anything about the quality of the poop or manipulating the quality of the poop to see whether the development is affected by what's in the poop.
Cool, thank you.
But I can tell you something that's interesting. A paper that just came out not long ago, I think. I don't know how many of you have heard about this bacterial infection in humans called Clostridium Difficile. I don't know if you've heard about this, but Clostridium is a bacterium, and I don't know if we have time to talk about that, but it's really interesting to see how we humans have dealt with this. Clostridium is a bacterial infection that basically will give you awful diarrheas. And if you're an older person or an immunocompromised person or a very young person and you get Clostridium, you're in bad shape. This Clostridium, particularly if you go for surgery for appendicitis into the hospital, you may end up coming out of the hospital with this Clostridium infection. Okay, in the hospitals are loaded with Clostridium. And the problem with this bacterium is that it is completely resistant to every single antibiotic that we have in our arsenal. So nothing kills Clostridium. And when you get infected with this thing, it destroys all the other healthy microbiome that you guys happen to have in your gut. And so it becomes a uniculture of Clostridium that you cannot get rid of. So what we have come up with as humans is this idea of doing transphonations of poop from your family. They request your family is healthy. The family gives poop and they make this milkshake of the poop of your family. And I know this is disgusting, but it works. This is the only thing that works. They do these fecal transplants where you basically get all the good microbiome from your family members. They used to have you drink this through a sonda. And so they would rephonate you through a sonda in your gut and then let it go. and all the good microbiome that you're getting from the feces of your family would actually kill Clostridium. Today, I think there's actually a company called Repopulate. What they're doing is they're actually growing this microbiome of healthy humans into these flasks, and then they're using a zonda to go the other way, a colonoscopy, to release all that good microbiome into your gut, which again finally gets rid of Clostridium. So, you know, termites seem to have been doing this for millions of years, but we are only now learning how to do this. And it appears that there's some humans that have a particular gut microbiome that makes them super donors. They're awesome donating at this microbiome that then really makes people who are infected with Clostridium really get healthy very quickly. So it's a combination of the type of microbes that you have in your gut that seem to be making you a super donor for these infections, which is pretty cool. So we should learn from the termites. They've been doing this repopulating for a long time. It's a good name for a company.
I think that would be an awesome way to end the second seminar series, talking about repopulating. Really fascinating stuff. I'll give just a moment in case anyone has a question that pops in their head. If not, I can stop the live stream and we can just have some social interaction here.
I had a quick
Go ahead,
question
Miles.
that's not necessarily pressing, but with the microbiome transfer, those super donors, do you know if that is something that's innate or if that's something that's like associated with their diet?
I'm not sure. Probably, as everything else in life, it's a combination of the two things, right? You have a genetic predisposition to have certain pH or certain acidity or certain whatever, plus what you're eating, and a combination of the two things probably makes you a super donor. Yeah, I'm not sure. I haven't really kept up with I just have the visual of the image of this human intestine, all very happy because it has all these microbes in their gut, and this makes you a very good donor. So hopefully none of you will ever need to be repopulated.
I don't see any more questions in the chat, so I'm going to first thank you, Becky, for a really, really remarkable,
thank you for having me.
Talk. And thanks to everyone for being here and for having another great series. For those of you who have to jump off and leave, we hope to see you in the fall. And